it looks like we have a good quorum here, so I think I'll start things off and say welcome to everybody. Thank you for tuning in tonight. This is the Upper Anacostia Summer Water Quality Program, a volunteer program, runs from May through September, doing a variety of sampling along Sligo Creek, Long Branch, and Northwest Branch. And it is sponsored by Chesapeake Bay Trust in Montgomery County, administered by Anacostia Riverkeeper, and with participation by Friends of Sligo Creek and Neighbors in Northwest Branch. We're looking forward to hearing analysis of the data. There's some interesting things that I think have come up and a lot of observations from a variety of folks. So we'll first be listening to Maureen, I think, and Petra, both, or one or the other, from ARC, telling us about what they found during the summer from our wonderful volunteers. And then we'll move on and hear from Neighbors of Northwest Branch about a little bit what they're seeing on the Northwest Branch. Then I'll talk a bit about our monthly program, and Dean, I hope, will help me out with that, looking at some of the data and trends we're seeing. And then we'll hear from Rachel Gauza with Parks, who's going to tell us a bit about the biological findings they're getting, particularly in Sligo, but on some of the other streams, I hope, as well in the county. We'll wrap up, I think, about 8:15 or so. I think we have time until then, Christine, if I'm correct. So thank you again all for coming. And I think, why don't we start? Maureen, are you ready to go?
Yeah, I think we're ready to kick off. So Patrick's going to share some slides for us, but I'm Maureen Mitchell. I work for Anacostia Riverkeeper, managing the water quality programs that we run. We run a slew of them throughout the district where the river where a river ends, but we also move all the way up with our monitoring program throughout the upper side, which is the Maryland side. And then Petra, who works here, I think I'll do most of the talking for today, save her voice a little. But me and her are we work super closely together and share this work. So everything I'm saying is coming to both of us. So we'll focus today with some results from Sligo Creek and Northwest Branch, but know that we do have a whole slew of information on the wider watershed that we'll be working on a report coming out, hopefully by the end of next month. Give us about 30 more days. We're hoping to have that out and published for you guys to give you a bigger view of what's going on watershed wide. So most of this was already covered, but the specific two projects we're pulling our data from today to present, both are CBT funded. Our Montgomery County has a wonderful slew of projects, including Sligo, Friends of Sligo Creek, and Neighbors Northwest Branch, Defensoras, Nature Forward. We also have partners UMD, but we are going to have present one little tidbit of data. We have a Northwest Branch site on our PG side, so I want to make sure we ensure that our partners in that are represented as well. Because this project would be nothing without all of our volunteers and partners working with us. So big picture, you can see on that right-hand map, all of the sites we're monitoring throughout the summer, all across the watershed. So there is, I believe, I can do the math. I can do math. It's okay. We have about 19 total sites that we're monitoring. 5 of them are in Montgomery County. 5 of them are in PG County. And then the rest of them are run by DOE and our DC project in partnership with Alliance for Chesapeake Bay. And that's 176 miles that watershed compromises. And about 60% of that is up in Maryland. About 30% of the watershed is in Montgomery County in that Sligo Northwest Branch area. And the Anacostia River has a long history of pollution, including sewage and stormwater runoff. And all these pollutions, so monitoring of these rivers and these streams are really, really critical. We've seen a ton of improvement in the green infrastructure in the area, and that has corresponded with some improvement in water quality. But we want to keep monitoring that and keep using this data point to push for more infrastructure improvements and more water quality improvements throughout its time. So obviously I just covered most of these. This is one of my favorite slides I like to show whenever I show you a presentation, just to remind us the entire breadth of issues that are facing any of these big watersheds like we have that are in these highly developed areas. Some of the things that we're seeing are things we are directly monitoring. That lovely dog pooping might be getting some E. coli in our water. We know sewage drains are also happening as well. We do have some nitrogen pollution coming from our fertilizers. We have trash. We have more climate change leading to more stormwater. We have salts, hopefully not too cold a winter this time, so maybe not too much salt this winter. But these are all challenges facing the Upper Anacostia watershed that these monitoring programs that we are just one of many are really critical to keep an eye on to create that baseline. So when those pollution incidents happen, we have that data to respond to. So specifically, when I present the data that Anacostia Riverkeeper is collecting, the three big things we're looking at are our fecal indicator bacteria. We are measuring E. coli, which is a really wonderful indicator of all the fecal and kind of the disease-carrying bacteria that might be in that water. And so that's the big one we're going to talk about 80% about, but we also collect data on turbidity. That's how much sediments and how clear that water might be. We collect pH data, how alkaline or how acidic the water might be. We get conductivity and dissolved oxygen, really important ecological parameters to kind of indicate if that water is healthy for plant and animal life. And we worked this year with partnership with someone from the Smithsonian Environmental Research Center to get some really great database on nutrients, nitrogen, phosphorus, and organic carbon. And so by the numbers, we had in Montgomery County, those were 10, sorry, in both Montgomery and PG County, those were 10 sites monitored across 200 weeks, looking at all the little vials of water, because each volunteer is going to be giving us about 5 each time. That was over 1,000 bottles of water that got processed by all our wonderful volunteers this year. We had 33 new volunteers that came out for the first time this summer. It was really great to meet them and get them trained. And we had 12 returning volunteers really continuing to kill it and getting these data points for us, with 35 total active monitors that were going out multiple weeks throughout the summer. So we have a really, really amazing and involved volunteer base that this data is all thanks to. So I think we're going to get to the fun start now. Here's some start to look at the data. So the image right here, these little pie charts you're seeing are percent passing. And again, we're focusing on that 80%, that E. coli data. So if something that's passing is saying that that bacteria level was low enough that according to the EPA standards, we're recommending that it's okay to make recreational contact at 410 units of bacteria or less. And so you can see there are quite a few sites in Sligo Creek in Northwest Branch that are doing really great, and that especially in dry, non-rainy, not a ton of stormwater conditions, are passing and meeting that recreational standard 70% of the time. So shout out to SC3, that's a Sligo Creek up north of Wayne Ave, as well as Burnt Mills on Northwest Branch. Both of those hit that standard 70% of the time, and those are excellent candidates. If you want to take your kids out and go splash in the water, if you're someone who might get a little hot and want to cool off and splash some water on your face while you're running, if it's in dry conditions, those are great places. But we did find some sites that are quite the opposite story. That includes Long Branch, which is a tributary running into Sligo Creek, that only passed 20% of the time. And so that was really showing us that that site, we have some concerns, and I would definitely recommend not making recreational contact without water. SC1, which is our middle, interesting enough, our middle site, that also didn't fare so well, only passed about 20% of the time as well. A lot of construction going in that area. It's very, very tightly packed residential with a lot of little tributaries. We might want to look into figure out where that might be coming from. And then down in Prince George's County, Northwest Branch 3, a new site we monitored this year. That one also only passed about 20% of the time. So a lot of really great sites, great recreational areas, but we also know some are not doing so well, and a little more work needs to be done to figure out what's going on in those locations. Ecological parameters, that's dissolved oxygen, conductivity, nutrients, those actually fared really well this year, both in wet and dry conditions. We did not have a single point this year that did not meet our ecological standards, which means it would be, they generally are in good. There was definitely a lot of fluctuation, and some are erring on the high side. But most of those other conditions, including bacteria, honestly, really are tied to how much rain had fallen recently and how much that stormwater is washing into the watershed. So let's zoom in on a couple sites a little more, or a couple of parameters a little more closely. This is going to be your E. coli data. It's really similar to the map we just saw, just showing it graphically here as well. You see, I guess you can't quite see my mouse, but I'm trying to point over here, NWB3, that's on the far right. That's your Burnt Mill site. And you can see that those sites are I'm so sorry. NW3 is the PG site down on the bottom. And then we can also see Long Branch. That's on the far left. I was getting those two mixed up. Those are two sites I was telling you are Long Branch and our 38th Ave, which is our site in PG on Northwest Branch. Even in dry conditions, those are frequently failing above that 410 level. Those are sites we're seeing a lot of concerns with. But you see Northwest Branch 2, that's the second from the right. That's our Burnt Mill site that even in our dry conditions, we were seeing double digits, under 100 units of bacteria, colony forming units. So really, really great data. Looking at the table at the bottom, you see that some of our sites are definitely improving across time. We only have 3 years of data, so this is really limited. We've had a lot of weather changes in the past 3 years that might be driving a lot of this trends, but we do see that things are trending up. These are percent passing, how much of the year that water was safe. And we see that across all the sites, we're generally seeing an increase, that sites are passing more often and water quality is improving. These are those two ecological parameters I was talking about, are dissolved oxygen. That's how much oxygen is available kind of in that water for any kind of macroinvertebrates or anything in that water to utilize. And then conductivity, it's a really measurement of how much salts are in the water. If conductivity is too high or too low, those salts, those minerals aren't available for growth and for the animals. We did find that conductivity and dissolved oxygen, again, every single sample I took this summer were in passing rates. Those were below what we have the EPA showing as a healthy stream. But there was still a lot of variation, both geographically and mostly what we saw was described by rainfall. If it was wet conditions, meaning it's rained more than like 3 inches in the past 72 hours, we saw that our conductivity levels went way down. Extra water just dissolves those salts, disperses them. And then we would see the oxygen levels go up after rainfall because we have more rain, the water's moving faster, mixing and pouring over rocks, especially at burnt mills. We would oftentimes get completely saturated 100% because there's that dam right before burnt mills that gets a lot of oxygen in that water. But nothing this year was concerning, but really good baseline data looking forward to anything that might happen in the future. This next one is some data that was given to me from our Rebecca Hale. She works at CERC and is running their urban watershed. So they're on year 1 of what we hope to be a multi-year project. And she gave me this from these graphics from a presentation she did to some peers. And so I want to make sure these are a little dense, but I wanted to show you them because they are really interesting. These are actually looking at county by county. The red would be those Montgomery County sites, so mostly our Sligo, our Long Branch, and that Burnt Mills. We do have 5 sites in Prince George's County as well. And what we saw was, in general, the data, the more rainfall they had, we actually would have the nitrates and the phosphates level going down, probably similar to conductivity. More water, the amount of nitrogen just kind of disperses more, and that level is going to go down. I will caution you, looking especially at that phosphide data, your gut is going to say, what's happening in Prince George's County? Why it sits so low compared to what we're seeing in Montgomery County? It happens to be that we sample every other week. And every other week, when I sampled Prince George's, it seemed to pour. And so we had a lot wetter conditions in our PG samples. That one, Northwest Branch 2, some of what we're seeing, Northwest Branch 3, sorry, our new Northwest Branch site this year in PG, we saw a lot of concerning data, high E. coli. A lot of these things might just be an artifact of the fact that we sample on opposite weeks. And the PG weeks, for whatever reason, whether God's told me it was going to rain a lot more those weeks. And so PG did seem to have a lot lower nutrient levels, but it mostly was because of that stream flow rate, that it was raining more often when I happened to sample at PG. So those are the big picture trends. Definitely happy to talk more if you guys had questions on any specific things. And again, we are working right now to report a really comprehensive report that will cover all of the sites. But I want to briefly remind you guys that this is a multi-year project and we are working on coming back for year 4. So we're currently applying for funding and expanded funding. We're writing a little bit bigger grants this year to have a little bit bigger projects in both counties next year. What this means is adding more sites, getting more data, and starting to pinpoint some of those locations that we saw were really high and try to figure out why. So of interest to you guys, we're adding some Northwest Branch sites in both counties. One is going to be kind of between Burnt Mills and our 38th Ave Park. I believe it's called Lane Ave or Lane Park. I'm forgetting the name, but there's a splash pad right there. Kids love to get in the water. So we're going to sample there. Lane Manor, I believe it just came to me. And that's kind of close to Riggs Road where there's current construction. So it is of interest to us. We're going to add a new Sligo site. This one is near and dear to my heart because it's on Dennis Ave. I live there and my kids love to jump in the water there when I'm at the playground. And my kids are not the only ones, but it will also give us a more upstream vision of what's going on Sligo Creek to start to understand the trends as you move downstream. And we're going to add in our Long Branch site. Our current Long Branch site is kind of on the upper half of Long Branch where the water tends to be more stagnant. So adding one a little more downstream where the flow is a little bit more consistent might help us have a better glimpse. Stagnant water is not the healthiest. And oftentimes we'll have increased E. coli. So having a little bit more of a high flow site might help us have a better sense of what's going on. And then we're also looking, hey, if we find more funding, if we find anyone who wants to sponsor sites, we can always add more. The bacteria monitoring is pretty pricey per sample, so that's always our limiting factor. We're going to keep working with CERC, keep doing that nutrient watch, hopefully for our volunteers, not including the syringe this year. If you volunteered, you know what I'm talking about. That wasn't fun. We're adding salt watch to our data set, so we'll actually have chlorine levels throughout the summer. So super excited for that. I know a lot of you guys do stuff during the winter, but adding it in to our summer program will help just complete our portfolio. And then I'm really excited about these 3 letters. We are adding MST next year. MST stands for microbial source tracking. We know there's E. coli in the water, but MST will allow us to know where it's coming from. So we're working with Professor at UMD to figure out, is the E. coli at that Long Branch site that's so high? Is that human E. coli, dog E. coli, bird E. coli? And by identifying where that E. coli is coming from, kind of what animal that E. coli's guts lives in, will help us have a better idea of where it's coming from and therefore how to address it. So really excited for that new partnership. It was pretty groundbreaking. So that just came out last Friday. So I'm personally really excited about it. And it will be throughout the summer, not every site, but I'm hoping to get at least a solid half, if not 2/3 of them. So definitely some Northwest Branch and Long Branch and Sligo Creek. I'll get some MSD data for you in about a year. Those are my presentations. I don't know, Pat, if you just want us to ask any questions you have our way, I see a bunch in the chat, but I'm also willing to just hold questions to the end if you prefer.
Yeah, I think we're going to do Q&A at the end. So everybody, if you just hold, go ahead and write down what you've got in mind and then add it to the chat or just, raise your hand when it's time for Q&A. Let's do it that way so we can keep rolling.
Awesome. Thank you, guys.
Yeah, sorry. That was excellent. Really happy to see all that data coming in, especially, nitrogen, phosphorus. That's great. MST for next year should be really helpful to know where this is all coming from, because that's something I think WSSC did 10 plus years ago, and it's been a long time since anybody looked at that. So I'm very pleased to hear you're doing that. Excellent. Well, let's go ahead. I think next we're going to turn over to Neighbors of Northwest Branch and just let Nora talk a bit about what they're seeing there. So Nora, go ahead and take it away.
There we go, unmute. Hi, my name's Nora. I'm president of the board of Neighbors of the Northwest Branch and have been for about 1.5 years now. I still feel super new. But today I just wanted to share, what we've been working on this year, and also just looking back over the year so far, what sorts of events or issues kind of stuck out to me, thinking from a water quality lens. So first, what has Neighbors been up to? Of course, we help these monitoring efforts with Anacostia Riverkeeper. That's why we're here. And our members find that information super valuable. And it's a great entry point for getting folks engaged and volunteering. We also have started a couple committees this year. So we have a cleanups committee. We've always done cleanups. We're just trying to expand our efforts and expand our participation in it. And we've also created a non-native invasives committee. They have a sort of project right now at Burnt Mills, which is one of the water quality monitoring spots, trying to return to that same spot over and over again and totally clear it out all those non-native invasives, so that we save not just the trees, but also the biodiversity that's there. And then something I'm really excited about is Salt Watch, as Maureen mentioned. That's going to be added to the repertoire next summer. So this winter, we're actually kind of teeing up that effort by starting it early. Salt Watch is a really easy citizen science activity. It's kind of like a pH strip, so it's a little bit less technical. And we're hoping to bring folks into our world of water quality monitoring that way, and also increase the history of sampling at those same sites that are going to be then sampled for water quality monitoring next year. If anyone here wants to get involved with any of that, if you're not already, please reach out. There's a contact form on our website, which is neighborsnwb.org. And then, yeah, just thinking about this summer and this year, what's been going on in our area. We've seen, unfortunately, a train derailment spilling plastic in Hyattsville that happened just last month. As I think was all over the listservs, there was an incident of the bus dumping their waste into a storm drain in Sligo Creek, something we're all obviously concerned about. Another thing is the air quality alerts from wildfires early in the summer. You know, I'm not sure, I don't know how that directly impacts our water quality, but it impacts our abilities to go out and recreate safely. It impacts the ecological functions of you know, the trees and the ecosystem all around our streams. So that's another kind of threat that we're facing. Even though those wildfires aren't happening here, they're still affecting us, right? And then a sort of ongoing issue has been the suffrage point development in Hyattsville. It's been discharging sediment. Sustainable Hyattsville is bringing lawsuits and doing a lot of really good advocacy work about that we've been tracking and supporting. And then in terms of good news, Prince George's County passed a plastic bag fee. Really excited about that. And Montgomery County passed a leaf blower ban. That one's really recent. But I'm looking forward to those sources of pollution reducing, hopefully, thanks to those laws. And then lastly, just sort of some big picture thoughts. We are in a time of unprecedented change because of the climate emergency. There's the MDE extension puts out these climate reports after each month. And I was looking back on them and I noticed everything is like, nothing's average anymore, right? It's warmer, it's drier, it's wetter, it's colder, it's back and forth. And as we saw, those storm surges are what's impacting our water quality. But also as like monitors, as people who pay attention to the reports, it really kind of surprised me. There was a week that was very dry. There wasn't any rain before monitoring, which normally we think, oh, it's going to be good. The bacteria is going to be low. Nothing's been washed into the streams recently. But then it turned out we had bad results that week. Maybe because it was so dry that things got stagnant. And so that sort of just stuck out to me as an adaptation, a change, something that's unexpected. And I think that unfortunately in this time of climate change, those are going to continue to happen. But as always, our work, our stewardship in the watershed is enriched by our feelings of connectedness. So getting together to talk about these things, and also feelings of responsibility. You know, the people here are the people who are the ones out taking data and using those syringes, even though it's tough, and stuff like that. So thanks for being here. Yeah.
That's great. Thanks very much, Nora. It's great to see for us, I think, too, since we're not in Northwest Branch very often, but it's interesting to hear what you're seeing there and what's happening with Hyattsville, how that's affecting what you're seeing in Northwest Branch too. So thanks very much for doing that. I think it just helps build a picture for all of us of Long Branch, Northwest Branch, Sligo, what's happening all around the area in the upper Anacostia. So very helpful. I'm going to go ahead and try to share my screen here. If that'll work. Okay, can you see that okay?
You might want to go present mode or full screen,
Yeah,
but we can see it.
okay. Let's see. Okay, can you see that one all right?
Yep.
Okay. So just stepping through briefly, I think what's helpful too is that the summer program is bi-weekly, you know, from May to September. Our program's every month, more or less, and all year round. And so I think it will help in some ways just to see some different kinds of data that is full season and is a variety of different parameters that hopefully complements what we're doing with the summer program and what you're seeing in Northwest Branch. So this program has been going on for about 11 years now, and we currently have 12 monitoring sites along Sligo Creek. We do a variety of parameters, pH, temperature, conductivity, surfactants, chlorine, chlorides, copper, etc. And we do share our data each month with DEP and Parks to let them know what we're seeing along Sligo. This is a quick look at the 12 sites that we go to, starting at the where Sligo daylights up by Blue Ridge Avenue up in Wheaton, and then continue on down all the way almost to the PG County line at 13th Avenue. We're about, I think, maybe a quarter of a mile from the PG County line down there. And what we're seeing a lot of, and Dean, please feel free to jump in at any point, but I do think something that we have seen for years is sediment, which luckily we're looking at with turbidity now with the summer program. But this is something that we measure each month at all of our sites. And it is certainly, as you can see from the pictures and from our data, an ongoing issue from construction sites, particularly the purple line at Wayne Avenue and downstream from there. But as you all know, it can be from a variety of construction sites, can be from lawns, people redoing their homes or redoing their yards, all sorts of sources. And it does have a very destructive impact, certainly on the stream bottom in particular. So that's something we're hoping all of our folks are just keeping an eye on and reporting whenever they can. In addition to sediment, another thing has been salts, in other words, conductivity, chloride, etc. That is certainly something we see. And Friends of Sligo Creek will be doing its third winter with the Isaac Walton Salt Watch, which we found to be very helpful. Our volunteers are excellent. They're very enthusiastic about it. So we're looking forward to doing that, and very pleased to see that we're going to be adding Salt Watch to the summer program as well. So I think it'd be wonderful to get year-round coverage to get a much better picture of what's happening.
If I could just add,
Yeah.
We got a number of unreasonably high salinity readings early this summer at several of our sites. And it was things that were consistent with or higher than we had seen after road salting events in the winter. And we're not sure why, but it's probably important to note that unlike the Riverkeeper that goes out every 2 weeks, no matter what the weather, we only test more than 48 hours after any measurable rain. So we're never seeing the turbidity loads that you get after a rainstorm. So when we do see turbidity, it's from something else, construction or whatever.
Yes. Yeah, good point, Dean. It is a dry read for us. That's just our protocol. But you're right. We did have some heavy spikes there. Perhaps because of the drought, those sort of flash droughts we had at certain points in the spring and early summer. We also look at coliform all year round, and unfortunately our repeat offender for years has been the Wayne Avenue outfall. So we think that does have a heavy influence on SC1 and also everything downstream from there, at least for maybe a half a mile or so, maybe a quarter mile. But we do that, and I will do a quick summary of some of our data a little bit later. And then another issue that we've seen, sort of a quieter one, but is surfactants, so detergents. There seems to be kind of a baseline of detergents in the background of the stream at all times. And has some strange cycles where it doesn't necessarily peak in the summer when you might think so, when people are, perhaps washing cars and the suds are ending up, down into the gutter, into the stream. We see often spikes during the winter. January and February can also be very high in addition to May and June. So it's a puzzle at points, but it is definitely an issue in some of our stretches of the creek and in some of the tribs that feed it. And I'll just take a quick look at some of the trends and numbers that we've been seeing from our data. The good news is, I would say, honestly, for most of the pollutants or parameters that we're following are tending to be steady or even trending slightly down at most sites. So that is actually heartening to hear. And I think that's encouraging, because sometimes we focus on what's going wrong and the problems, but there does seem to be at least a plateau, and in some cases improvement in some of these parameters at many of the sites. That said, chloride, I think we are seeing that rise at most sites. Bennington Drive is particularly afflicted with chloride almost all year round. It drains downtown Silver Spring, so that may account for much of that. Wayne Avenue is also high, and Flora Lane, which comes off of Georgia Avenue, commercial area, also tends to have a high chloride level most of the year. Upstream is noticeably lower, which is interesting. Above the Beltway, those sites tend to be lower values. But again, the trend is up with both chloride and free chlorine. It's interesting about what we call central, which is the end of Long Branch as it enters Sligo. Typically our highest for free chlorine fairly consistently. And Bennington, which is so heavy in chloride, is actually our lowest when it comes to free chlorine. Again, the trend is up for chlorine. It's also up generally for coliform, but specifically E. coli. Often our total coliforms seem to be trending slightly down, which is an interesting divergence, but E. coli tends to be slightly moving upward in many of our spots. Turbidity is very heavy where purple line is going on, as you would expect at Wayne. But it's also heavy where Sligo daylight's way up in Wheaton at the Blue Ridge site. That's one of our highest for average, mean turbidity levels. So that was a little surprising, but interesting. Conductivity, again, Bennington and Wayne, they are draining a lot of downtown Silver Spring, so they have a tough route when it comes to conductivity. And I would say surfactants, again, Blue Ridge is interesting because they're very high there. Detergents tend to be, it's sort of a slow, very stagnant body of water, and that may account for some of that. And it is where the Sligo Creek first daylights out from beneath impermeable surfaces. Our pH varies from about 7.5 at Wayne to 8.33 at 13th Avenue. So it does seem to pick up. The values get more alkaline as we continue on down from daylight on Wheaton all the way down to the PG County line. I would also echo something we heard before too, is that we're tending to see variability of our parameters of our readings is increasing somewhat. We're seeing bigger spikes, whether it's in air quality, whether it's in temperatures. The highs are higher, the lows can be lower or at least the same. So that is something I think we're starting to see over the past 2 or 3 years in particular. Certainly the wildfires accounted for a lot of that when it comes to air quality. We did see big spikes in PM10 and PM2.5 during the summer, as we would be expected. And I would say, what else? That's pretty much, I think, sums it up for that. I wanted to quickly talk about Water Watchdogs, which is our program to try to catch pollution before it happens. It's a wonderful program run by Ann Vorse, and just want to put in a plug for that. Anyone can help. Very easy to go ahead and report through our site or the Water Watchdogs tag. And there is still ongoing pollutants. Folks mentioned the commercial bus that was emptying sewage right into the creek this summer. We've had firefighters using foam that went down directly into the creek. And we do have many problems with, could be drywall skimmings, could be washdowns from, it's not clear, washdowns from a construction site, maybe it's paint, but there are certain sites, particularly Flora Lane, that in that area seem to be getting hit very consistently with this sort of thing. So we encourage everybody to please keep an eye out on the creek and do what you can. And this is the site to report to if you do see something. Yeah, and I think that's it for me for now. So I will go ahead and stop sharing and go ahead and pass it on to Rachel as soon as I can. Rachel, are you there?
I'm there. Hi, Pat. Hi, everybody.
Yeah.
I am waiting to share my screen. I have you cannot start screen share while the other participant is sharing. So I might need you to click one more button, Pat.
That's probably me. Yeah, give me a second here.
No problem.
You should have a button in the top in the middle that says stop.
Yeah, I'd like to find that button. I seem to have lost all control. Apologies. Yeah, all I'm seeing, unfortunately, is the view. I see all of you.
I don't think there's much troubleshooting I can do on my end, unfortunately.
Here we go. Yeah.
Okay, that's looking
Yeah.
promising.
All right.
No
Yeah,
worries at
covered.
all.
Here we go.
All right. Okay, well, good evening, everyone. I appreciate the opportunity to be here tonight to speak with you about some of the work we do in the resource analysis section and specifically through the biological monitoring program. So Montgomery Parks is working cooperatively with Montgomery County Department of Environmental Protection and others to monitor our waterways and assess stream conditions using biological indicators. So we follow specifically the Maryland Biological Stream Survey protocols, which involve spring visits to sites to collect benthic macroinvertebrates using the DNet that you see there, where we evaluate 20 square feet of representative habitat in a given 75-meter station. We then return to those same locations in the summer to examine the fish community. We use those benthic macroinvertebrates, sorry. We use those backpack electrofishing units that you see there, which temporarily stun the fish, allowing us to efficiently collect them, examine them, and return them to the water at the end of the survey. And during both of those visits, we are also evaluating physical habitat parameters of the stream, as well as some basic in situ water chemistry. parameters, so some of the same ones that Maureen was mentioning that are of significance to those ecological communities. So pH, dissolved oxygen and concentration and percent saturation, conductivity, temperature. And then we're also, while we're making these data collection visits, collecting other supplemental data. So we do amphibian and reptile surveys, as well as observations of ephemeral wetlands and non-native invasive plants. And we take all of these data and combine them in order to create a holistic view of our stream health, both in our parks and beyond. So it sounds a lot like critter counting. It's a bit more complex than that. We bring our spring stream bottom samples back to the lab where they're subsequently subsampled with our organisms being identified to genus. The fish, as I mentioned, are evaluated on site. We take aggregate weights at the end of each of our electrofishing sampling passes. We will identify each individual to species, and we also examine each for anomalies prior to release. So what you're seeing here is a golden shiner out of the Wheaton Branch portion of Sligo Creek with some black speckling on it, which happens to be black spot disease that's caused by a trematode. It's a very common and widespread parasite that we see on some of our animals, and we see them in greater proportions in more stressed systems. So that's one metric that we use to help look at the overall biological health. In fact, we have several representative metrics that all those various identifications and counts go through. Those are then translated and tabulated into a score of index of biotic integrity for both biological communities. So you'll see that BIBI for the benthic in macroinvertebrate index of biotic integrity score, and FIBI or fish index of biotic integrity score. And then those can be further examined together as an average stream condition in which we can then assign a narrative of excellent, good, fair, or poor. So that gives us a really good digestible snapshot at a location at a given time, but we can also parse things back out into those individual IBI scores, since our two biological communities are telling us slightly different things, and then also break it back down into some of the metrics and look at how they relate to one another and try to better understand aspects of community structure and the function behind those scores. So the data between Montgomery County DEP and Montgomery Parks have been collected since the mid-1990s at roughly 800 stations across the county. As of 2023, there are 15 stations in Sligo Creek watershed that are being monitored by either agency. For example, some of these like dots in blue are some of DEP's current and historic monitoring sites. Many of those are up in the upper portion Sligo Creek and in some focal areas such as Brewood tributary. And then Montgomery Parks has adopted or established several sites in Sligo as well that are monitored at varying frequencies. Typically, biological monitoring is conducted in 5-year intervals, but if we are focused on certain areas that are either of interest for restoration or that were good candidates for restoration and we've made changes to the stream channel, we'll look at those at different frequencies. We'll get baseline information and then generally years following restoration, post 2, 4, and 10 for subsequent biological data collection. So you can see Wheaton Branch up there that I referenced earlier, Edgevale tributary over there towards the midpoint of Sligo Creek watershed in Montgomery County. Then we also have special investigations. Those are highlighted in yellow, and they're kind of much more even variable than what I've described, kind of more on an as needed basis, or when there are certain needs to fill in or promote continuity of different data sets. And then we have annual monitoring stations, the one in Sligo Creek down towards the Prince George's County line, and as the name suggests, monitored every year. And that's the one I wanted to kind of focus on this evening, as well as leave some time for discussion. So Montgomery Park's annual monitoring station is near the Carroll Avenue Bridge. You can actually see it in that photo here. And it falls between Carroll Avenue and Jackson Avenue. This portion of Sligo Creek main stem is considered a third order stream, and our 75-meter station is located upstream of the confluence with Long Branch tributary. As you can also see in this picture, our 75-meter study area is heavily channelized with large riprap. It's wide, it's fairly shallow. Our most recent measurements have it averaging about 9.5 meters in width with a maximum pool depth of 60 centimeters. We also, of course, take into consideration the catchment areas or the drainage areas to our biological monitoring stations. And in the case of this particular station, SCSC 314, we're looking at a drainage area of about 7.4 square miles, which is roughly 2/3 of Sligo's total 11.1 square mile drainage area. The station was established in 2000 and began to be monitored annually starting in 2005 as part of a cooperative study with the Metropolitan Washington Council of Governments to kind of help expand a larger look at trends in the Anacostia River watershed. So here are those index of biotic integrity scores. We have our fish plotted in black and circles, and our benthic macroinvertebrates plotted in gray squares here. Both have been normalized and expressed as a percentage, so they're a little bit easier to compare. And when you combine them together across 2000 to 2023, we see that the trend for this portion of Sligo Creek main stem rates as overall low fare across that full depth and breadth of monitoring years. When we look at the two communities that we focus on separately, though, we see that the fish index of biotic integrity typically ranks higher and shows quite a bit less variability than we see with our benthic macroinvertebrates overall. Again, two communities can be informative because they're responding differently. By nature, fish can move in and out of a station, for example. Bugs are going to be a lot more sessile and sedentary. And then some of our benthic macroinvertebrates are extremely sensitive to certain things, such as road salt and other pollutants, while fish in general may be a bit more hardy. And so when we split those two communities apart, we can start to get a better idea of what is going on and what is kind of influencing that overall low fare across all the monitoring that we have available so far. So here's looking at those scores through kind of a different lens. This time we have fish plotted in blue. We have the benthic index of biotic integrity scores in red, and then that combined score of both in purple. You can see that all are showing an upward trend across the data that we have, albeit much more gradually for those benthic macroinvertebrates. The fish community, however, is probably the most impressive there. It climbed the most. It's also driving that combined or average stream condition up quite a bit. Some of that can be attributed to some increases that we have seen in sensitive taxa. Specifically in 2016, we began observing small numbers of northern hogsucker that you see pictured there. That's a pollution intolerant specialized species that feeds on aquatic invertebrates. And we've continued to see that species every year since, including 2023. Small numbers, but some differing size classes and age classes, which is pretty encouraging. Benthic macroinvertebrates, on the other hand, do kind of continue to straddle poor and fair, even in some of the more recent years, but they have achieved a fair ranking in 2023. That particular sample actually also included a microcaddisfly that we do not detect very often. In fact, we only have a handful of records from main stem Northwest branch over that very comprehensive data set since the mid-1990s countywide. So they were observed, the taxa itself was observed in Sligo for the first time this spring through our monitoring. It was one individual in a subsample, but it's noteworthy not only because of that kind of more uniqueness, but it is one that has a moderate tolerance to pollution, and it also feeds via scraping algae and diatoms off rock surfaces where they're spinning their cases. This one is pictured without its case and a little stretched out. So like the microcaddisfly, the organisms that persist and their associated pollution tolerances and ecological roles tell us quite a bit about water quality and stream health. And the fish community in particular here at Sligo is telling an encouraging story. Here we're looking at different proportions of the various pollution tolerances within the fish community. And if you look really close towards the top, you can see blue bars. Those are representing sensitive species, in this case, the northern hogsucker. And we've seen in more recent years, from 2016 to 2023, that the fish community score is actually ranking high fare and on the cusp of achieving an overall good ranking. It did successfully pass into good from 2020 to 2022 at Sligo Creek main stem, again below kind of the Carroll Avenue Bridge area. This is in part due to some of the species diversity that's supported there. Since 2016, we've seen 10 species or more supported, and across that time band, more in the range of 12 to 16 species of about the 25 fish species that we would expect in the watershed. Beyond just the species diversity, a large driver behind this kind of high fare to good fish community that we're bouncing between is that proportion of species with intermediate pollution tolerance that you see represented in orange here. That includes species such as American eel and the long-nosed dace that is pictured here. Long-nosed dace are also highly specialized feeders that are known as riffle-benthic insectivores. They rely on stable, rocky, fast-moving habitat and a variety of benthic macroinvertebrates to eat. So substantial portions of them can be found in healthier stream systems. So what we're seeing for 2023 and kind of that slip back down into fare isn't too different from what we saw in 2018. That's the two are fairly similar ratios broken out. So basically the counts of some of those moderate individuals and the specialized feeders are being down a bit and replaced more by those tolerant generalist species represented in purple. So to summarize all this, Sligo Creek truly is a sum of its parts and its surrounding watershed. Biological monitoring is allowing us to do a stream health checkup. Streams are dynamic systems by nature, but when we see large deviations from the norm, we're basically looking at a sick stream. In the case of at least this portion of Sligo, and as it kind of reflects the overall watershed condition, or at least that 2/3 or so of the watershed upstream, we are seeing kind of a disproportionately unbalanced and shifting biological community. And this is symptomatic of a stressed system. This is largely the result of just a large catchment area and the extensive urbanization therein. There's, of course, multiple historic and ongoing influences and both kind of chronic and discrete stressors related to that. Certainly dating back to, when the Sligo Creek Parkway was even constructed in the first place, but, think back to some of those ongoing stressors and the problematic pictures that Maureen presented on her slide, some of the pollutants that Pat highlighted. They're all working together cumulatively to have an influence on the biology in that system. So with that in mind, there are likely limitations to what we can expect the biological communities to achieve, but I think we can certainly celebrate Sligo's resilience. The positive trends that we've been seeing since 2016 suggest that there's at least sufficient habitat and water quality to support a pollution intolerant fish species. And at time, we're seeing great densities of individuals with more moderate tolerances to pollution, and that includes the benthic macroinvertebrate community as well. In fact, looking at our fish again, the American eel that you're seeing a video snippet of there was at its highest in 2022. And Sligo Creek is capable of hosting some really large and long-lived individuals. Sligo has really, come a long way in the past few decades. It's exciting to see biology and habitat really starting to reflect that. I think it's safe to say that the water quality and conditions are much improved from the conditions dating way back and certainly in the early 2000s. Some of that can be attributed to things like green infrastructure, as Maureen mentioned, and the tightening of stormwater controls to help reduce that volume of stormwater, sediment, and non-point source pollutants rushing into the stream and changing the dynamic of the stream flow and chemistry and habitat availability. The reasons for improvement are numerous, and they're not always easy to isolate. But another thing kind of worth calling out or giving a nod to is the ongoing WSSC infrastructure and rehabilitation efforts to repair failing assets and try to protect those that are at a greater risk of failing due to just the age of the infrastructure and their locations. Some of that work, for example, that's more recent is up in Sligo Creek SVU 5 as well as SVU 2 near Shoaler Lane. Parks is invested in that and other improvements. We work with WSSC as much as we can, as well as other agencies as much as possible to ensure the best outcomes and the best decisions are being made. And we're also working as hard as we can independently for opportunities to protect and enhance aquatic habitats. This is involving a variety of techniques where we're trying to improve stream channel habitat, stormwater outfall stability, creating floodplain enhancements and various protective measures. So I had to highlight a couple amphibian ambassadors of those efforts. We have thanks to some of the efforts of our Montgomery Frogwatch community scientists, some supplemental data on amphibian breeding in different portions of Sligo Creek watershed. This particular wood frog that was cited was part of a community that was heard calling and breeding in a couple created wetlands along Sligo Creek Trail near Nicholas Drive. And then to the right of that is a northern two-line salamander that was seen having colonized a channel stabilization and habitat creation project at the Edgevale tributary to Sligo Creek that I referenced earlier, along with tessellated darters. So they found favorable habitat once that project was complete, and we'll continue to monitor out there as well. But perhaps I would say the most important part, here to talk about parks, of course, but I really want to acknowledge that Sligo is fortunate to have dedicated ambassadors and stewards like all of you. Again, Sligo is a sum of its parts, and you're very much a part of it. Montgomery Parks is out there. We're doing what we can to help support others, as well as what we can do to treat excess stormwater, to try to address and heal things that are causing sick streams and prevent problems in the first place. But we're looking for folks like you to help spread that knowledge and increase awareness of all of our collective efforts as we work towards protecting our natural resources. So I'm really happy to hear the results of your effort tonight, and thank you for hosting me.
Thanks very much, Rachel. Great to see that side of it. It's a wonderful compliment to the chemistry and bacterial work that we were looking at from the summer data with Maureen. Thank you very much for showing us the biological side and also reminding us too the connection to the land habitat as well, that it's all connected system and what we can do from there. So thanks so much. I think at this point we're at Q&A. We probably have about 15 minutes or 20 minutes for questions. So if you would like to put something in the chat, or if you want to go ahead and use the raise a hand for reactions, either route will work. Please go ahead. But thank you to all who've presented and given us a lot to think about with regard to Sligo and Northwest Branch and Long Branch. Nora, did you have something?
I do. I have a question for Maureen. From early in your presentation, I loved seeing that map of all of your monitoring sites. And I wondered if do you have a place where you share that map, like with the results?
That
Um,
map is part of a report, which we will, we are working on putting together. It's very much in draft form, but that, I don't think, Petra, do you think we'll have it done by, give us 30 days, a month, and we'll have that report? Maybe, definitely by, let's say, Thanksgiving, we will have that report ready. It'll be available on our website. We'll share it heavily. There'll even be printed copies. We're trying to get some kind of bilingual version out there. So it is something that Petra's amazing and whipped up for this presentation, but it will be definitely highlighted and will be all over our social media and our webpage as soon as we get it done. It's a lot of data to look at.
Gotcha. Yeah. There's I just throughout the season we see like the Prince George's County sites and the Montgomery County sites kind of isolated because it's the every other week. So
Yeah.
I just really appreciated seeing them all together.
Yeah, and we recognize, and I think looking at that nutrient data set, especially for logistic reasons, doing them on opposite weeks has made a lot of sense for ARK. A lot of moving pieces, but there obviously is, specifically think about Northwest Branch, which crosses. There's some drawbacks too, because we don't have the same sample from the same week of the up and downstream sites. So we're brainstorming solutions moving into the new year, because I really would like to have a better full picture of what's going on up and down. If we get the samples at the same time, we can draw those better, and at least making sure we're making maps and images and graphics that allow people to see the watershed as a whole.
Thanks, Maureen. Michael Wilpers, did you have a question?
Right, thanks. A really excellent panel and amazing statistics and effort by everybody. I'm wondering if it's sort of a two-part question on the same topic. Are the all the different water quality parameters that everyone's measuring are benthic organisms and fish susceptible to the same, or are there a different, suite of insults that, the specific ones are really bad for macroinvertebrates, but fish can deal with it, or vice versa? Or is there just one set of, like the two that are the most deadly to both sides? How does that shake out?
Sure. I could take a stab at that. So both sets of biological communities are going to be reflective of the cumulative stressors. So they're all going to be sensitive to those variety of parameters that everyone is kind of looking at. There will be varying sensitivities among them, both within the communities and as opposed to both communities. So we do know, for example, that our measures of conductivity are associated with declines in benthic macroinvertebrate community scores, and that certain taxa within the benthic macroinvertebrate communities are highly sensitive to road salts and are lost. Some of this, again, can be a little hard to tease out. So for example, where we see very low conductivity readings and support some of those very sensitive stonefly and mayfly taxa, for example, are also in watersheds with very high forest cover and very low impervious cover, and less of the issues with stormwater. But there are some strong correlations between the toxicity that occurs with conductivity and road salts and metals, for example, compared to others. Likewise, we see some interesting shifts with nutrient enrichment. So certain benthic macroinvertebrates will all of a sudden kind of flourish and take over in response to high levels of different nitrogens and phosphorus-based pollutants, whereas others maybe not so much. And then we'll even start to see some fish take advantage of those conditions as well. So it's always kind of a little bit of a game of cat and mouse too. When we're looking at both benthic macroinvertebrates and fish, sometimes we'll see something like the response to a drought conditions, kind of the spring after the summer is like that. So it's trying to figure out, okay, what's telling us what? This summer it was probably the fish, and then, okay, now we're seeing that effect in the benthic macroinvertebrates. So I think it's very useful to be able to have some of these other parameters, and especially in locations that we're not actively looking at, like our annual monitoring site is kind of down towards the bottom of the catchment, but starting to identify these hot spots like, oh, here's a problem for E. coli. Oh, Bennington Drive is a hotspot outfall for this pollutant, and this pollutant, I think, can help give us a better understanding of what the biology is reflecting.
Great, thank you, Rachel. Also, I do see in the discussion, there's a fair bit of question and discussion back and forth about heavy metals, microplastics, some of the breakdown products from tires for cars and trucks. Does anyone have any data or want to speak to that as far as how that affects the system, the aquatic system generally?
Oh.
Or
I
not?
do, yeah, I mean,
that's okay.
I am certainly not saying one way or the other, but in terms of like what we're able to evaluate thus far with our biological monitoring data, I'm not sure that that's something I can
Right.
speak conclusively to.
Yeah, okay, just thought I'd throw it out. It does seem to be of
Mhm.
interest to folks. There's another question too about some beavers setting up a new spot, perhaps a new dam near the Sligo Path in the soccer field.
Hmm.
Question about how does their presence relate to water quality and threats to trees?
I can briefly take that question on.
Okay.
In terms of the water quality, trees are a little outside my expertise. You know, beavers are warm-blooded, meaning that they do have E. coli themselves. And we know that beavers can have other hosts of associated pathogens, like Giardia is most frequently going to be transmitted by beavers. So, you know, it's a natural impact on the water quality in terms of bacteria, but we do expect that that would lead to an increase. We had a site down in DC that actually Watts Branch had some beavers living in it this summer. So different water area of the watershed, but we saw the impacts of Watts Branch. Normally, it usually has high levels, but skyrocketed when that beaver was present. Also, beavers are going to lead to stagnation of the water. That's the whole point of their dam building. So if they're building that dam, that's going to stagnate the water, which would oftentimes lead to other things increasing. That stagnant water is going to decrease in that dissolved oxygen. We might see some increases in conductivity as those things pool. That stagnancy is going to allow those E. coli that the beavers are putting in the water to grow even more. And so beavers are natural, you know, so it should be a natural thing. That's why E. coli levels are never going to be zero, because we have warm-blooded animals living in the streams are going to be getting that in. But I'm actually really curious. I might go walk that area tomorrow and try to find them for myself. But if they're setting up close to a monitoring site, it definitely would lead to us having some higher in that area. And I would definitely recommend not recreating directly downstream of a beaver dam or in the damming area itself, because those areas will have higher bacteria levels and lower water quality that I personally wouldn't want to get in contact with. The trees, I'll let someone else take.
Thanks, Maureen. Good advice. Rachel, do you have anything to add as far as the entire system with trees and the riverbank?
Sure. Well, I don't know that I can really speak for the trees either, Lorax style, other than to say and emphasize that yes, beavers are a natural component of our ecosystem. So it's interesting as we talk about like different ways in which we measure water quality and the lenses we look at for water quality. So what it may mean to recreate may be a little bit different than what we would look at from an ecological perspective. So beavers are excellent ecosystem engineers. They're very good at creating habitat and specifically aquatic habitat in their just normal behaviors and need to dam and stop flowing water. They end up creating ponding, impoundment, and wetland habitat. Wetland habitat, of course, hosts a variety of different other organisms that would benefit from it. So we get an expanded amount of macroinvertebrates, additional fish habitat, maybe some different conditions favored by other fish, as well as the opportunity to do what wetlands do so well, which is help manage and control stormwater. So we do sometimes think of like, oh, beavers and flooding, but really in creating the wetlands, they're offering opportunities to slow down and hold water to hopefully eventually, as things start to normalize, settle out some of those things. So I'm usually a bit of a beaver believer. Of course, we have to manage that with, if it's going to flood out a soccer field, maybe our wildlife ecologists need to take a closer look at it. But we see them offer some ecosystem services, and there's even some interesting local research and investigations out there looking at techniques that beavers do as a stream restoration approach. So kind of mimicking the things that beavers do in order to promote overall water quality.
Excellent. Yeah, that would be interesting, giving them a spot to work and see how that affects the entire system. There's a question here from Michael. Are high levels of E. coli equally detrimental to fish and benthic organisms as they are to humans?
Yeah, so E. coli is going to be living in, it's going to impact warm-blooded creatures. So fish, benthics, those actually aren't going to be able to get sick from E. coli, since E. coli attacks and lives and makes a mess of your intestines, which those cold-blooded creatures won't have. However, we do use E. coli as indicators. So, it's an easy one for us to test. That's why we're testing that one. But when we do see, and studies have shown that when E. coli spikes, other pathogens are likely spiking. So while the E. coli itself isn't going to be harming the benthics or the fish, when we do see spikes in those conditions that allow E. coli to grow, it is highly probable that other pathogens, ones that might be attacking, and I don't know a ton about pathogens that attack fish, but I've seen plenty of studies that show that there is high correlation. So when we have prolonged E. coli spikes, sites like Long Branch that are just constantly failing and maxing our systems out, I would be highly suspecting that other pathogens that might be dangerous to fish or to benthics would also be present. But E. coli itself, warm-blooded only. So dogs, humans, cats, birds, don't let them get E. coli, but other guys aren't going to be impacted.
I would note that one of our testing sites at Fosk is also on Long Branch, but down at the mouth, only about 50 yards above where it runs into Sligo. And we do not usually see high coli there.
Yeah, I think that's, we're going to add a second Long Branch site. I actually want to talk to you and Pat and figure out if we could almost duplicate that exact same location. That's always going to get us better data, because it'd be really interesting to see up downstream. We're taking samples within an hour of each other. I do think that higher stream flow, we're going to see those E. coli levels lower there. It's the upper Long Branch where it's really stagnant. The higher levels have been seen, but time will tell.
Yeah, it'd be an interesting follow, because the chlorine levels there, as I mentioned, in Long Branch tend to be very high, and the coliform levels tend to be very low. So maybe a causation there or not, but it would be interesting to do a little more digging there and see what we find.
Yeah, I think the addition of that source tracking that Maureen mentioned is going to be really exciting, because if we can maybe more conclusively say something like, oh, that might be human, then we can start to trace up the WSSC sewer infrastructure and be like, oh boy, there's leaky pipes, or there's this type of issue. Otherwise, with someone like the benthic macroinvertebrates, what we might expect, or benthic macroinvertebrates in particular, is the E. coli, if it's from like an animal source, for example, you usually see nutrient enrichment go with it, and kind of that shift in biological communities as we look at some of those individual metrics. So certain things like black flies are like yum, where it might be a little bit more toxic or prohibitive to other taxa. So that's interesting. We do have 2 biological monitoring sites on Long Branch as well. Oh, let me think where best to describe them. We were last out there at one of the sites in 2021, which was that was our downstream station that is below Piney Branch. I'm trying to think how low or how far below Piney Branch.
Like where the Terrace Elementary School is, or?
I think it's further than that. And then we have an upstream station that's near Long Branch Wayne Local Park in like Garland Avenue area. And when we looked at some of the historic data, I mean, overall Long Branch is quite poor. In fact, historically has only supported one fish species, the black-nosed dace. However, in 2016, we did see the benthic macroinvertebrates towards the upstream station, again, near Garland, in the fair range. And then we took a look at the downstream station in 2021, following a pretty substantial water main break, just to kind of see what was happening. And the benthic samples were dominated by non-biting midges and things that are pretty indicative of a struggling stream system. Whereas we did happen to see 3 fish species. So there was kind of a expansion in that shifting system. So black-nosed dace again, but also white sucker and creek chub. So all very tolerant and pioneering species, but pioneering nonetheless. If you've only had black-nosed dace, hey. And
Right.
then another portion of Long Branch, we have observed American eel, where some of that stream work was going in near Haddon Drive. We'll move fish out of the area of impact, and there were eel in that area. So be interesting to see if American eel, as we continue to focus some efforts on Long Branch, if they're one of those species that might tell an interesting story.
Yeah.
I think hearing all this really shows the importance, and I'm so thankful, Pat, you put this together, because there are so many monitoring programs in our area. Obviously, you have yours, we have ours. The Parks and Rachel does stuff. I know Nature Forward does some benthic work alongside a lot of us, and like DP has gauges. And so just as we continue to grow and think about future years of all of our various work, making sure that this data is, however we can, put on layers of maps and visualizations on each other, because I think we could see some really beautiful correlations and start to put a more powerful story together than any of us individually can with just our singular data set. So these conversations are really beautiful.
Agreed. Yeah, it'd be great to start coordinating and seeing if there are patterns that start to show up as we bring the data together and overlay it. Michael, did you have a question, Michael Wilpers?
Yes, speaking of Long Branch, I think you mentioned there was considerable stream, I don't want to call it restoration, but a lot of work done on Long Branch between Piney Branch and Carroll. And I'm wondering if any monitoring downstream from that would be from the playground at Houston, I think it is, downstream to Carroll, or even from Carroll to the confluence, if the completion of all that work has resulted in, noticeable improvements downstream.
Yeah, I think for those, time will tell. So yeah, it's a little difficult to refer to them as stream restoration from an aquatic ecology perspective to some degree as well. A lot of times we're opportunistically trying to do some advantageous work in association with other things that are happening. So WSSC needs to get in there and tweak things like, what can we do from the aquatic ecology side to make improvements? A bridge needs to be replaced. Like, what can we do to stabilize and promote better habitat? That said, we've been largely relying in Long Branch for those 2 stations. I think they also were established with data collection in 2000. I would have to go back and double check, but again, other ones that were pretty long term that helped to highlight. So we'll continue to look at those 2 stations at varying frequencies in order to try to connect some of those dots that you're mentioning. In terms of Long Branch, there's certainly tons of other initiatives and a lot of interest there in Long Branch. So that may also inform the next time Montgomery Parks gets out there, though I could see us getting out in maybe the next 2 years. We only, despite saying, oh my gosh, 800 sites across Montgomery County, obviously both DEP and Parks has a limitation in where we can get and have to hit that strategically and have our own kind of individual goals. So for Montgomery Parks, it's about 24 sites a year that we can get to on our parkland. So we kind of have to play a game of Jenga of who's going to go where and when, and make sure we also hold true to the continuity of those like long-term annual sites.
Right. Working with the resources that you have.
Name of the game for everybody, for sure.
It is. I actually want to talk. I wanted to ask a quick question, actually, really for probably Maureen and Rachel, but just because y'all looking at other streams, we tend to be focused basically on one, it's with some tribs. But are there other physical analogs, streams that are similar to Sligo in the area that you could think of that you can talk about that are either have substantially better or worse, Phoebe or IBI scores? I mean, I'm just curious how it rates compared to some of the other streams and watersheds in the area. If anything comes to mind.
Sure. Yeah, so maybe I can talk a little bit specifically Montgomery County, and then Maureen, if you want to fill in anything with like the DC perspective, I think that would be great. Very broadly, in terms of Montgomery County, we see kind of the trend of down county stations, or those that are closer to DC and kind of those suburbs, having impacted biological communities compared to those that are up county. A lot of that is very closely tied to the amount of impervious surface that's in those watersheds. Maryland DNR has some very strong relationships. They show the Center for Watershed Protection, where there's like an understanding of once you go above 10%, you're looking at impacted stream systems, as I mentioned, earlier with our Sligo Creek annual monitoring site, we're looking at 30%. So stuff that could be ranging in like the non-supporting threshold for biological communities. But we can kind of reverse some of that via things like green infrastructure and stormwater controls. Now, can we get it back to what it would have looked like with 3%? And before those sensitive taxa, or the most sensitive taxa, were lost? Well, that's a question I think all of us practitioners are trying to really put our finger on. So yeah, all that to say, broadly, down county watersheds have these kind of shifting, unstable biological communities, impaired biological communities. Cabin John, for example, Northwest Branch to some degree. We do have an annual monitoring station in Northwest Branch. We're just starting to really start to pull that data apart even more. We looked at it some in 2020, so I owe another kind of more detailed dive, I think, like we just did for Sligo a bit. In the case of our Northwest Branch annual monitoring station, which is below Oakview Drive, we have a fairly stable fish community at our location. It's actually better than Sligo's. I think that may be a bit of a function of the topography that's out there. Pretty expansive riparian buffer, particularly on the right bank that we have at that station. But the benthic macroinvertebrate community is kind of right down there with Sligo and hits some of those variabilities. So we'll be taking a closer look at that. And then Little Falls, I would say, is a watershed that is maybe looking a bit like Sligo may have a couple decades ago, with some of its stressors and where the biology needs to go to start trending a little more upwards.
Great. Thank you, Rachel. Maureen, did you want to add anything?
Yeah, I think it's a big question, right? Because I'm looking at the watershed wide, and we're looking specifically, the data set I'm super used to is the recreational data set. And Sligo is a little unique, both in that we have the most data from Sligo, as our partnership is so strong, and a lot of the other streams we really have singular grabs on. But what we really do see across the watershed in that bacteria data, it's very interesting. Like our uppermost Sligo sites, Burnt Mills, thinking of Northeast Branch and Paint Branch, we actually grabbed a couple of samples from this year. Those really upper fingers of tributaries tend to be really good. We see E. coli levels, they're still impaired. We're still obviously failing at 20-30% of the time, but it's good. You get down to the bottoms of tributaries, as I talked about that new Northwest Branch site on 38th Ave. Some of our other tributaries that we do test that are a little bit closer to the main stem, a little bit more in the densely packed suburban area. Not that we're not super suburban here, but not quite in the city yet. But we start to see those ones really start to fail right at that. We have like Hickey Run in DC, kind of near the Arboretum. We have Watts Branch and a lot of our PG tribs. Those ones are lucky that we do see bacteria levels really spike in those areas. And then as you move downstream, talking about the actual river, The Upper Bladensburg, which is one of our sites in PG, that one also doesn't fare so well, even though we have a lot of water there to help. You would think it would dilute it, but all the stormwater rushing in at that point from all those little tribs going through all those industrialized regions, that's going to spike up. And as you move towards the bottom of the river, close to the confluence of Potomac, we're talking about Navy Yard, Kingman Island. I'm sure you all saw we were advertising for our swimming event this summer. That was unfortunately rained out, but that shows, and we see data there, that when that water gets that big, and we've had a lot of really amazing sewer infrastructure work happen in the district, the bacteria levels kind of are back down, and we're passing 80%, 90% of the time on those sites. So it's interesting. The very, very top of our watershed and then towards the bottom of our watershed, we're seeing really low bacteria levels, but it's kind of that middle area where we have those smaller streams where green infrastructure really, and the stormwater is really impacting it. The MS4 systems that have a lot of leaky connections. That's where a lot of the work is. And Sligo isn't generally, although Wayne Ave and that outflow for the SC is definitely problematic. I think in general, it's a pretty unique stream, and the bacteria levels are really low and very high. And that has to do with the advocacy work that's been done to really help Sligo start to rebound compared to other streams that don't have as strong advocates and have not that green buffer to help keep them clean. Those ones tend to be a little bit higher.
Great, thank you. I appreciate both. It's just helpful to have that overview because you see a lot of other streams and waterways that we don't really have a chance to look over. So it's helpful to get perspective that way. We are coming down toward the end of our time, actually. Does anyone else have a question or two? We've got about 4 minutes left or so.
Well, I'd like to ask, Pat noted that FOSC sends its data to Parks and DEP, and I'm wondering, what does Parks do with the data we send them?
It is definitely something that we appreciate and we continue to keep an eye on. Most recently, we try to look at it in areas of interest. So it's a little hard for us to use it like fully holistically at this time. But for example, Long Branch, again, is one of those areas of having a lot of interest, different resources coming its way. Pat and I corresponded about like where the monitoring locations were, what was being found in Long Branch, and like making a point to say, okay, sure, here's what the biology is. Here are some things that could be happening, and here's some additional data on top of it that helps support some of that trend or some of the things that we already suspect out there. So it's really helping to fill in the gaps for us at Montgomery Parks, which is very valuable as kind of the land stewards themselves of being responsible and having a little bit of weight to try to tick things in the right direction and not just be like, can't do anything, not our land. Like, no, we should really be making our best efforts to do so. I can't speak specifically for how Montgomery County DEP utilizes the data, though I know, at least for Little Falls, that it has drawn some really important attention to what could be going out there that is leading to such concerningly high levels of E. coli and unsafe conditions for recreation.
All right.
Thanks.
Thank you.
Thank you.
Yeah, we've had an ongoing conversation with DEP too on the other side about Wayne Avenue, and we're hoping at some point to get some closure and have them take a look at that deeply and track down those sites of coliform, which we've seen very steadily for many, many years. And I think that will certainly.
Hopefully, all of the sewer work they're doing in conjunction with the Purple Line construction, and apparently a very large sand filter they're building right at Wayne Avenue. Let's hope that when those things are commissioned, it'll make a big difference. I mean, that old outfall always has high E. coli, but it has very low flow most of the time.
Yep.
When we test, 48 hours after a rain. So my guess is when it rains, there's lots of E. coli being flushed out of there.
You're probably right, yeah. We'll see what we can find out with DEP, and it would probably change what we're seeing, certainly in the summer program, if that were to finally be addressed. I think SC1 and SC2 would do much better, but we'll hope the DEP gets a chance to look at that in more detail. Well, I should actually wrap us up. I want to say thank you again to Maureen and Nora and Rachel very much for your terrific presentations. Really a lot to think about. Thank you, everybody, for attending and for helping out. And we'll look forward to seeing you out there, hopefully with volunteer programs of any sort that appeals to you and along the various waterways that we all get to enjoy. So thank you again for coming. Have a wonderful night. And we'll look forward to seeing you soon.
Well,
Take care, everybody.
Thank you,
Bye-bye.
everyone.
And have a great night.
You too.